Welcome to part 2 of this session. We've done the first part of nucleic acid. We introduced what nucleic acid are. Excuse me. We also looked at what make up nucleic acids, the components of the nucleic acid. We looked at the difference between nucleic acid and the nucleosides. Nucleotides and nucleoside. And we also discussed the formation of the nucleic acid, like the structure, how is it formed, the condensation reactions which take place, and we also discussed further on the types of the nitrogenous bases, where we said we have the purines and the pyrimidines. When you're talking about the purines we said there are two types of purines. We have the adenine and also the guanine. When you're talking about the pyrimidines we have three types. We have the uracil, we have the thymine, and also the cytosine. So we went further to look at the structure of adenosine monophosphate, where we said it has only one phosphate and it's made up the adenine and also the ribose sugar. We went further to look at the structure of adenosine triphosphate, where we said it is made up of the nitrogenous base, which is the adenine group, and also the three phosphates, the triphosphate. The tri means three, so three phosphates in the structure, and a pentose sugar specifically, which is called the ribose sugar. We went further to look at the structure of guanosine triphosphate. We said the difference between guanosine triphosphate and also the adenosine triphosphate, the only difference is that all the nitrogenous base, in guanosine we have the guanine, in adenosine we have the adenine. So this is what we discussed in the previous session, part 1 of this video. So this is the part 2. We pick it up and make now the concept into details. So let's now discuss the structure of the DNA. So this is where we ended. We looked at the ribose sugar and also the deoxyribonucleic sugar. We looked at the ribonucleic acid, the deoxyribonucleic acid, and all that. So let's continue now. We need to understand one important thing here. What we need to understand is that this DNA we are talking about, or we've been talking about rather, it is a double-stranded molecule. Okay? Or should I say helix? It's a double-stranded helix. And this theory of being double-stranded, okay, it was proposed by Watson and Crick. So Watson and Crick proposed this theory that DNA is double-stranded. So we need to understand that. So what does it mean when we say DNA is double-stranded? So it has two strands, of course. Double-stranded means it has two strands. So that is the structure of DNA. Let's have this. So it is made up of two strands. So it's like that. That is the structure of DNA. But when you're talking about the RNA has only one strand. This one is single-stranded. This one is single-stranded molecule, okay? So what we are going to have for RNA will just be that. Maybe it will just be thick enough like that. So this is rRNA. It is just a single-stranded molecule. Now let's look at the structure of DNA. We are saying it is a double-stranded molecule, right? So even the nucleic acids there, they will be bonded in such a way that they form two strands. Okay? So let's have a certain, let's have this structure. Then we have the phosphates there. Then there we have the phosphate. This phosphate is bonded like that. This phosphate again is bonded to that. Okay. Then what we need also to understand is that the strands in DNA, they are bidirectional, they are antiparallel. So I'm going to invert this. So this side, of course, we have the nitrogenous bases. Let's say that is the adenine. Nitrogenous bases, let's say that is the thymine. Nitrogenous base, let's call this one as the cytosine, right? So the other strand will be inverted to be like that. Okay, to be like that. Then we're going to have the phosphate there. Like that. The phosphate there. Like that. The phosphate there. Then we're going to have the nitrogenous bases. But before I can even pair these nitrogenous bases, I want you to understand one important thing, even before I pair up this. So what you need to understand is that the adenine makes a bond with the thymine. So the adenine there will correspond with the thymine. Then the thymine there will correspond with the adenine. The cytosine there will correspond with the guanine. So this is what you need to understand. So for the adenine and the cytosine, it forms two hydrogen bonds. There is a double bond. Okay? Then for guanine and the cytosine, there are three hydrogen bonds. So we can say that the bond between the guanosine and also the cytosine is much stronger. Why? Because there are three hydrogen bonds between the guanine and the cytosine, but there's only two hydrogen bonds between the adenine and the thymine. And one thing, excuse me for that. So one thing you also need to understand is that these strands are bidirectional. So if we have the 5' there we have the 3', right? There we are going to start with the opposite. We'll start with the 3' end there, then we'll end with the 5' end. Okay, so let's look at another concept here. Let's look at another concept. So the best thing we need to understand is that the purines, they pair up with the pyrimidines. The purines pairs up with the pyrimidines. That is very important concept to understand. You can't have the purines being paired up and also the pyrimidines being paired up. You can't have that situation. So always the purine must be paired up with the pyrimidines. And another thing I want us to understand here, it is very important to understand that in rRNA we said there is no what? There is no thymine, right? Thymine is only found in DNA. So if we are talking about the rRNA, what will be paired up with the adenine is not thymine. Okay? It's not thymine. Nevertheless, what will be paired up with the adenine will be uracil, because uracil is found in rRNA, but thymine is not found in rRNA. So that is very important for us to understand. Let's look at the functions. Let's look at the functions of DNA. So what is the function? What is the basic function of DNA? So the basic function of DNA is just to store the genetic information. Okay? This one, store genetic information. That's the basic function of DNA. And we need to understand that, let's say it is storing the information for black hair. So what is going to store that information for black hair is what we call the DNA. And we also need to understand that DNA is the same for all organisms. Is the same for all organisms. It is just the same. So this is very important for us to appreciate. And let's look at now the function of rRNA. For rRNA, the function of DNA, we've said it is just the storing of genetic information, right? But for rRNA, this is the one now which transmits that information which is stored in DNA. So this one transmits information, of course this information is genetic. It transmits genetic information of DNA. But there are certain things which needs to occur as this information is being transmitted. There are certain things which needs to occur So let's look at what needs to occur before this information is transmitted. So we'll have, let's have maybe DNA being converted to DNA. So when DNA is converted to DNA, that process is called the replication process. So DNA being copied in another form of DNA, that is replication. Then this DNA will be converted to RNA, and to be specific, it is mRNA. So this now mRNA, from DNA to RNA, this conversion is what we call the transcription. So it is very important for us to understand that. So this is transcription. But there's also the conversion which takes place between RNA, mRNA to be specific, to proteins. What do we call that conversion? So that conversion is what we call the translation, right? That is called translation. So let's understand also the writing movement of complementary structures. Let's say you have 5 prime, then we have adenine, guanine, thymine, cytosine, then 3 prime. How can you write the complementary structure of this? So the complementary structure, the complementary pair of 5 prime is 3 prime. The complementary structure for adenine, we said adenine pairs up with thymine, guanine pairs up with cytosine, thymine pairs up with adenine, and also cytosine pairs up with the guanine. Then complementary pair of 3 prime, that is the 5 prime. So very much important for us to understand. Now, how is that the chromosome come about? How is the chromosome coming about? Because when you're looking at this DNA, it is just a small portion. DNA is just a small portion of deoxyribonucleic acid, right? So when you're talking about DNA rather is the deoxyribonucleic acid. But when you're talking about a gene is a small portion or small segment of that DNA. And we understand to say that for DNA, to exist, there must be some structures, right? There must be some structures which are known as the chromosomes. Chromosomes are much essential. So, what forms up, or how is it that the chromosomes are coming into existence? Because chromosomes come from DNA. That is one important fact we need to understand. DNA is what forms chromosomes. So there is a conversion. Let's say when DNA wants to form the chromosomes, it has to coil. Once it coils, it forms what is known as the DNA helix. Okay? It has to coil, then it forms the DNA helix. So that is very important for us to understand. So we are saying for chromosomes to form, this DNA has to coil. Once it coils, it forms what is known as the DNA helix. Okay? It forms what is called the DNA helix. Then this DNA helix also, it will coil. This DNA helix, it coils also. Once it coils, it forms what are known as the histones. Okay? So DNA coils around. Once it coils around, it forms what are known as the histones. Then these histones also will coil. Once they coil, they will form what is known as the nucleosomes. Okay? They will form what are known as the nucleosomes. Once these nucleosomes also coil, these nucleosomes will also coil to form what is known as the chromatin. Then this chromatin will coil also. In fact, this chromatin doesn't coil as such, it will just condense. This chromatin condenses. So we are saying the chromatin now condenses. Once it condenses now, it will form the chromosome. So this is how chromosomes are brought about. So it's DNA coiling to form DNA helix. Once that DNA helix coils again, it will form what are known as the histones. Now the histones again will coil. Once they coil, they form what are known as the nucleosomes. The nucleosomes coil to form chromatin, and chromatin will now condense just to form the chromosome. So we need to understand that. So when you're talking about the chromosome, that is the structure. So the chromosome will be like that. That is the chromosome. But when you're talking about the chromatin will just be like the chromosome cut on the middle. So this will be the chromatin. That is how a chromatin is. So it is very important for us to appreciate this concept. Okay? Very important. So let's have an example. Let's say you have DNA. It is being converted to rRNA. We said DNA to mRNA specifically, that is transcription, right? So let's say you have these pairs. So we have maybe 3', we have the adenine, we have the thymine, we have the cytosine, another cytosine, then 5' end there. So what will be, we call this to be a template strand, right? A template strand. We call this to be a template strand because it is the template strand that is going to code for information. Okay, so we have the template strand there. The template strand is the one which is known as the non-coding strand, right? Yeah, the coding strand is what codes for information. Okay? So let's have a template strand there. Then what will be the complementary base pair? So adenine will pair up with thymine, thymine will pair up with adenine, cytosine with guanine, cytosine with guanine, then 3' end there. Okay? Now this is a non-template strand. non-temperate strand. Now, what if you want to form rRNA? So, for rRNA, we use this non-temperate strand, which is also known as the coding strand. So, this coding strand is one which is going to code for rRNA. So, we don't have the thymine in rRNA, instead we have the uracil. So, we'll substitute uracil there, but we have the adenine, we have the guanine, we have the guanine. These 3' will be maintained like that. So this is rRNA. So to transcribe the non-coding strand into rRNA, you just remove the thymine, which is not present in the rRNA. Okay? And we said that the bond between the adenine and the thymine is much stronger than the bond between the cytosine and the guanine. We need also to always understand that. And when you're talking about the pentose sugar, we looked at the backbone of DNA, right? We said the DNA is made up of the backbone, which is the pentose sugar. And one thing you need to understand or to take home here is that this pentose sugar which is formed, it is a D-furanose ring. Because the D-furanose ring is able to form a ring, right? It is the D-furanose ring. So we need to understand also that. Okay? It is very important to understand. And we need to understand that the instrument which was used to determine the DNA structure was an X-ray. So in case they ask what instrument was used to determine the rRNA, it is, the DNA, it was the X-ray. Now we talked about the 3 forms of rRNA. 3 forms of rRNA. Maybe let's conclude by talking about the 3 forms of rRNA. So we have what is known as the transfer RNA, tRNA. So for transfer RNA, this one transports the amino acid. This one transports the amino acid. Then we have what is known as the rRNA. rRNA, which is also known as the ribosomal RNA. You see, the ribosomal RNA, this one joins with the proteins to form what are known as the ribosomes. So, forms the ribosomes after joining with the proteins. So, we need to appreciate that. Then we have the last one, which is also which is called the messenger RNA. So we have the messenger RNA, which is also called the mRNA. So what is it about the mRNA? So the mRNA is now the one which is able to carry the genetic information. Okay? So the mRNA carries the genetic information. And it also synthesizes the proteins. You need to understand that. Okay? So it translates the nucleotides to amino acid. That is the function of mRNA. So we've talked about the transfer RNA, that it carries the amino acid, and this is during translation into cytoplasm. Okay? We've talked about the function of the mRNA, which we are saying it carries genetic information which codes for protein. Genetic information or genetic code. The mRNA carries what is known as the genetic code. The tRNA transports or transmits the amino acid, and also the mRNA joins with the proteins, the rRNA joins with the proteins in order to form those interesting things known as the ribosomes, right? Okay, and the ribosomes, we have the 2 types, okay, of which we will look at in the next session. We have the 2 parts. Okay, so let's conclude by, and the other thing you need to remember here, when you're talking about the transfer RNA, this one is formed at the middle. It is synthesized in the nucleoplasm, okay? It is synthesized in the nucleoplasm. So that is very important. It is synthesized in the nucleoplasm, but when you're talking about this rRNA, the rRNA, this one is synthesized in the nucleolus. The middle of the center of the nucleus, right? That is the nucleolus. That's where it is synthesized. But for tRNA, it's synthesized in the nucleoplasm. Or the other name for nucleoplasm is simply the cytoplasm. Okay? So it is synthesized in the cytoplasm, in short. Then same applies to the messenger RNA. The messenger RNA is also synthesized in the cytoplasm. So it is very important for us to understand that. So this one is synthesized in the cytoplasm. Cyto. How do you spell cyto? So cytoplasm. Like that, right? So it is very important for us to understand this. And when you're looking at this, let's talk a bit about this mRNA. So let's talk just a bit, not even in so much detail, just a bit. So you can have this structure. Let's say it's like that. Then you can have the tRNA here. You can have the tRNA here. You can have the tRNA here. Okay. Like that. Okay. So this is what is known as the tRNA. And it has a lot of arms. It has the transverse arm. Okay. It has. Okay. Before you can look at that, we need to understand that the longer one between the two arms there's a longer one and the shorter one. The longer one is the 3'. That's where now the amino acids come to attach on the longer one, which is the 3'. Then we can have AUG there, which is the start codon, methionine, right? Methionine. So we need to understand that there are three base pairs there. So three base pairs form what is known as a codon. A group of 3 nucleotides is what gives a codon. This is the mRNA. Okay? Then that is the tRNA. So we've said that this tRNA has a lot of arms. That's a lot of arms. Okay. Then that's one there. This is the rRNA, the ribosomal RNA. So one thing you need to understand is that the codon is only found in the mRNA. That's where you can find the codon. Okay? So it's very important for us to understand that it is only found in RNA. Then this part of the tRNA, that is the anticodon. So if you have a codon there, that will be the and now codon. The codons which are going to be formed there. There will be anticodon, the opposite of the codons which are going to be formed or found there. So So this is just the basics of what I would want you to understand. Okay, just the basics. And to conclude this, you need to understand that the ribosomes, when you're talking about the ribosomes are made up of subunits. They are made up of subunits, which are also known as the sedimentation coefficients. They are made up of subunits. Okay, let's conclude by talking about this. They are made up of the subunits, or what are known as the sedimentation coefficients. So, one thing I need you to understand is that in eukaryotes, so you have the eukaryotes here and the prokaryotes or prokaryotic cell. So we have the small subunit, the large subunit, and the combined subunits there. So in eukaryotes, the small subunits, they are 40S. The large ones are 60S. The combined ones, they are 80S. But this is not mathematics, because if you say 40 plus 60, that won't give you 80. So it is not mathematics. Okay? It is not mathematics. So this is what you need to understand. Then for eukaryote, just subtract 1 from everything we've written there. So 40 minus 10, subtract 10, not 1, sorry. Subtract 10. So 40 minus 10, we'll have 30S. So 30S in prokaryote, the small subunit. Then we have 50S, 50 minus 60 minus 10, 50S for large ones, and 70S for combined ones. So it is also important for us to understand that. And this S we are talking about, it is just the sedimentation, the setback. Okay? The setback. It is known as the setback constant. So it's just the sedimentation rate. The rate at which these ribosomes are able to settle on the base, on the bottom of the test tube when you carry out centrifugation. Okay, so in a lab, you have a centrifuge, then you have a test tube, you put the ribosomes. After you put the ribosomes, then you carry out centrifugation. Okay, when you carry out the centrifugation, like you make it spin you make that test tube spin at a fast rate. So you discover that these things we are calling the subunit, the ribosomes rather, they are going to settle. Now they are going to settle at different rates. That is what we are calling the setback. Okay? So this information is very important. So this is the end of nucleic acid. If you found this video to be helpful, don't forget to like, share, subscribe to the channel so that each time a new video is uploaded, you are the first one to watch it and grasp the information. Thank you for watching. Yours is a good day.